Gautam sir, can you hear me, sir?
Yes, sir.
Gautam sir,
Yes, sir.
ମାନେ ପାର୍ଟିସିପେଣ୍ଟ ଆହୁରି ବେଶୀ ନାହାନ୍ତି ସାର୍ ଏବେ ଯାଏଁ 6 ଜଣ ହିଁ ଅଛନ୍ତି ଟୋଟାଲ।
हाँ, हम
ୱେଟିଂ
देखिए
ରୁମରେ ବି ବେଶୀ ପିଲା ନାହାନ୍ତି।
वही तो है। सर 8 से लेके देखा
हेलो सर, बोलिए। वो बाहर का है क्या? मैं उसको वेटिंग रूम में डाल दे। हाँ।
Sir, good morning.
Good morning. Am I audible?
Yes, sir.
Yes, yes.
Oh,
Sir, good morning, sir.
good morning.
Sir, welcome to Roland Institute of Pharmaceutical Sciences, and thank you very much for accepting our invitation.
Thank you.
I request our Honorable Principal, Dr. Ravi Kumar, sir, to welcome the resource person for the session of 6th March.
Sir.
Sir, Ravi Kumar, sir, please unmute yourself. Principal, sir.
Yes, sir. Good morning, Dr. Tapan.
I request our principal, sir, to welcome resource person.
Good morning, Dr. Tapan. I'm Dr. Ravi Kumar, Principal, Roland Institute of Pharmaceutical Sciences. Sir, please unmute yourselves.
Unmuted.
Yes. Good morning.
Good morning. Thank you for accepting our invitation, sir. And welcome virtually to Roland Institute of Pharmaceutical Sciences.
Thank you, sir. I request Dr. Gautam Kumar Jenna, sir, to introduce our resource person to the participants.
Pleasant good morning to one and all. Most respected resource person of 6th March of part session of Aicte sponsored short-term training program. Professor Tapan Kumar Gandhi, IIT Delhi, Honorable Principal Sir, Vice Principal Sir, and coordinator of this Sttp, Dr. C.H. Niranjan Patra Sir, co-coordinator Mr. Kanu Charan Panigrahi, my dear colleagues, and most respected delegates. I, Dr. Gautam Kumar Jenna, Associate Professor, Roland Institute of Pharmaceutical Sciences, feel honored to introduce Professor Gandhi. Dr. Tapan Kumar Gandhi is currently working as Associate Professor in the Department of Electrical Engineering, IIT Delhi, and Adjunct Faculty at IIT Jodhpur. He is also research affiliate to MIT USA. He received his PhD fellowship from MIT USA and obtained his PhD in biomedical engineering from IIT Delhi. Following his PhD, he has spent 3+ years as postdoctoral research scientist at MIT, USA. Dr. Gandhi was also awarded an Impaired faculty in the Engineering and Technology category of the Department of Science and Technology, Government of India. During this 5 years tenure, He is awarded as the Excellent Inspire Faculty by DST, Government of India. His resource expertise spans from computational neuroscience, brain imaging, assistive technology, biomedical instrumentation, machine learning, cognitive computing to artificial intelligence. He has published papers in top ranking journals like Nature, PNS, Current Biology, PLOS Biology, IEEE Transactions. He has more than 140 publications in the international journals and conference proceedings. He is PI and co-PI of nearly 20 crore funded projects from industry as well as Government of India organizations. He has received many awards in India, including one from ex-President Dr. Kalam and abroad for his academic excellence and outbreaking research. His research work is not only published in top-notch journals, but also appeared in many popular presses, including Time Magazine, the Boston Globe, the New York Times, MIT News, Harvard News, Wall Street Journal, and also in Science Magazine. Dr. Gandhi's work was selected as cover page in Science Magazine in 2015. He has delivered many invited talks in national and international universities like MGS, Harvard University, Yale University, MIT, Copenhagen University, University of Groningen. He is one of the international member to review European Union joint projects on vision science and collaborator in many international projects. He has 3 patents and 3 technology transfer to Ministry of Science Justice, Government of India. He is serving as expert member in various research committees in Government of India, UNESCO, as well as reviewers in many journals like IEEE, Elsevier, and Springer, etc. He has also received Platinum Jubilee Young Scientist Award 2015 by National Academy of Science in India and GYTY 2019 award. Presently, he is chairperson of Project Prakash Charitable Trust in restoring of vision and providing education to visually impaired in India. On my personal front, me and Professor Tapan are associated since our childhood. With this brief introduction, I request Professor Gandhi to address the gathering.
Thank you, Dr. Gautam, for this nice and illustrious introduction. Good morning, everyone. Instead of taking lots of time for all this formalities, we should start the talk, because actually today is a very hectic day for me, because after the talk I have back-to-back 2 meetings in Government of India. So that's why I'm thinking that I'll talk more than 1 hour, but I don't think that I'll be able to speak more than 1 hour. But the main goal of my talk would be to encourage you how we can do fundamental science to bring the breakthrough not only in science but also in translational technology. The title of my talk is Restoring Sight to Visually Impaired. So here, when I'm talking about visually impaired, you know that this is a very big problem in India. But if you look at the current scenario of India, you'll be surprised to know that everywhere we are talking about, can you see my slide? I changed my slide. Can you see it?
Yes, sir, I can see.
Some magazine cover, some beautiful lady. Can you see it, right?
Yes.
Yeah. So these are the
Yes,
snapshots.
yes,
I just actually I just Google and I found these are the snapshots that everywhere we are talking about. India is going to the superpower. India's economy is booming. Okay, maybe that's fine, but in the back door, you will be surprised to know that 1 in every 100 Indians is blind. The incidence of childhood blindness in India is 3 times that in the West. And you will be surprised to know that whatever the blind population in all over the world, India is the house for nearly more than 50% of the blind population. So you can imagine what might be the problem. I mean, problem in terms of socio problem, economical problem to the country like India. And these are the major causes of blindness. You can see here, corneal scarring. So you know that if there is any scare, I mean, there is any injury in the cornea, the person would be blind. Cataract. You must have heard about old age cataract, because you know that when somebody become old, they have very thick lens in their eyes, and this is called the old age cataract. But you will be surprised to know that many child also take birth with this kind of cataracts in their eye. And if they are born with this cataract, then they will be destined to blind. Retinal dystrophies. Retinal dystrophies means when the retina will be detached from the cornea and there will be no connection to see the things, right? Infections. Infection is another major cause of this kind of problem, the blindness, because you know that in India, nearly 80% people live in the village. And if you go to any government hospital, you'll see that it's not properly sanitized. Even many mothers in our country, they deliver baby in a very normal kind of setting, which is not sanitized, and the chances of infection is very, very high. And due to this infection also, the newly born baby also have the chance to be, to be blind. And then finally, the congenital rubella syndrome. And congenital rubella syndrome, you know, this is a virus. And for this, nowadays, you know that the mother, should be vaccinated when they're pregnant or when they're going to deliver a baby. But you also know that in the villages, nobody care about this vaccination. And due to this problem, there are the chances of congenital rubella syndrome. But you'll be surprised to know that out of all this kind of blindness, nearly 40% blindness is either treatable or preventable. But due to the lack of knowledge, lack of expertise, or lack of resources, we are not able to restore sight to this, even this 40% people, those who have a chance to see. So if you look at the consequences of this blindness, that why this blindness is a very big problem in the country like India. So if you look at this figure, I've just given you some figure. If you compare the lifespan of a blind child with respect to the counterpart, the control child, you'll be surprised to know that the lifespan of blind child is 15 years less than their normal counterpart. The childhood mortality is very high. Even 50% of the blind child, those who take birth, they don't get a chance to see their second birthday. That means they die. You know that in India, like country, when the child is born blind, the education prospect is also very, very less, right? That means they don't get a chance to go to the proper school, because we don't have this kind of special school everywhere in India. Maybe in very big city, you will find some, but even they are not up to the standard. And due to all this problem, the employment prospect is also very, very less, even less than 1%, right? Because when the child has not gone to school, and definitely nobody will give them the job. So that's why, due to this problem, every year, India is losing nearly 10 billion US dollar. That means this is a big loss to the country like India. So you might think that how big the problem is. Okay. I hope that you can see my slides. I'm changing my slide now. In my slide, one child with a red cap. Can you see it?
Yes.
Okay. So if my slides is not changing, please let me know. Otherwise, I won't be able to follow you. Okay. So the pressing humanitarian need is to provide treatment to curably blind children, to whom we feel that we can cure them or we can treat them. We need to provide them treatment. So there are different kind of blindness there. The blindness may be, as I mentioned to you, the blindness may be due to many different kind of factors. Even you must have seen in the train or in the bus that even, or maybe on the road, that many blind people, they don't have the properly developed eye, right? Even some people have, you know, that they're born all right, but after certain age, they get blind. We call it retinitis pigmentosa, RP problem. That means their photoreceptor cells get degenerative or they die, right? So here I'll highlight very fundamental problems which can be tackled by the scientist or by the researcher or the people from different background. So it's not about, I mean, you must be thinking that I'm from electrical engineering, IIT Delhi. How come I'm talking about biology? Yes, there nowadays, you know that in the new government of India education policy, the government is giving more emphasis on interdisciplinary kind of research. That means maybe you are in the pharma, or you may be pursuing your career in pharmaceuticals, but you need to have knowledge from everything. Let's say if I'll talk about AI is everywhere nowadays. And if you know how to write a code, how to apply your coding ability in computational biology, that would be fantastic. So that means you need to bring all different kind of expertise to your domain. Even you'll be surprised to know in my lab, one scientist who is from pharma background, she is working to address the problem of blindness in different way. I'll tell you, I'll just highlight you, but I don't think that I'll get a chance to tell you the story. I mean, what kind of work she's doing. Okay. So here, just I'm highlighting you that what we see when we are talking about blindness means this is related to vision. That's what we see. So you just imagine every day morning when you get up, you see this beautiful world around you. And people ask you, who is telling that this world is beautiful, or who is giving this kind of information? Definitely you will tell that maybe my eye can see this beautiful world. That's why the way my eye is seeing, that's why I'm telling this beautiful, right? But do you know who is responsible for this? This is not the only job of the eye, but this is the collaborative effort of eye and brain. Sometimes what happens, you can, the rays of light will pass to your eyes, but if your brain will not decode the information, that means you won't be able to see anything in this world. And that problem is called amblyopia. You must have heard the term called amblyopia. And in the layman language, it's called the lazy eye. That means you have eye, you have brain, you are all right. But sometimes in many people, there are many people who can see in one eye, but not in another eye, though the eye looks absolutely fine. Because maybe the eye is perfect, the eye is giving the information, but the brain is not able to decode or encode those information. So you can see here in my first picture, the left corner, you can see that this is the left and right eye, and it's coming through this, the information usually comes through this optic nerve, and then it meets in the optic chiasma. And here, through this optic tract, it usually go to this lateral geniculate nucleus, LGN. And from this LGN, you can see there are many fibers-like structure, which is being projected to the visual cortex of the human brain, right? And where the information get processed. Okay, so that's why you must have I mean, in Tare Zameen Par, you must have seen this movie. And that kid who is the main character of that movie, when the teacher asked him the question, I mean, ask him to explain something, then he said that jo dikhta hai wo hota hi nahi hai, jo hota hai wo dikhta hi nahi hai. And this really the right kind of answer he had given, because what we see, okay, this is what our brain usually show us. Okay, so you must have seen some illusion. So illusion is basically which does not exist, but we can see the illusion because our brain is showing us, right? So, but actually illusion does not exist, but our brain usually help us to see the illusion, right? That means what is happening, maybe we don't see the right thing, but which is not happening, maybe our brain allow us to see that what is not happening, right? So now we are clear that what we see, what we hear, all this sensory information, these are not the job of all the sensory organ, what we have in our body, but this is the collaborative effort or collaborative processing between eye or brain, ear or brain, or our smell bud with brain, right? So this is how they do. So I'm not going into details of all this thing. I'll just tell you big picture of all this thing, right? So as I told you, okay, this is you understood. So I told you one problem called the cataract. Cataract means in Motiabind. In Odia, it's called Motiabindu, right? And second one is called the retinitis pigmentosa. And you just imagine if somebody born with blindness, and if that person will be blind for the whole life, maybe okay, this is okay, or maybe fine. But just imagine if you are born all right. Let's say I am born all right since my birth. I've seen everything until age of 20. And after 20, if I'll lose my sight, how I'll feel? This is more tragic, right? So this is the problem called the retinitis pigmentosa. And in this problem, this is a hereditary problem. And here, what happens? the rods and cones, which is called the photoreceptor, that get degenerate, right? And that's why they lose their sight, right? So I'll actually, okay, here I'll show you at the end of my slide what exactly you can do being expert in pharmaceuticals, right? So the third thing is that, okay, so here, okay, I'm showing you here. You can see here, this is the eye. Left picture, you can see this is the eye. And inside the eye, the small section you can see here from the retina, we have taken the small section and which is being enlarged. And you can see here in the right picture, this is how the different layers are present in the eye, right? From the sclera to the optic nerve, you can see there are rods, cones, you can see sclera, then choroid plexus, that pigment, and then rods and cones. And rod and cones, these are called the photoreceptor cell. And they translate the light into some form and then pass to the bipolar cell. And then bipolar cell process this information and then pass to the ganglion cell. And this is how it goes to the optic nerve and then to the visual cortex. Just imagine in the retinoids pigmentosa, if this photoreceptor cell like rods and cones will degenerate, what will happen? So here my question is, can we send the same information even if we don't have the rods and cones? Can we substitute this process by using some kind of neural stimulator? So in order to bypass this layer, if this layer is degenerated, then can we bypass this layer? So we are using some kind of drugs like lidocaine, lamotrigine, to understand how this neural transmission happens. We are using different kind of recording techniques, how we can record the ERG, electroretinogram. So we are just trying to understand the behavior, and we are doing this kind of study on the rabbits and rats, right, with your drugs. So I'm not going to details. Maybe if I'll get a chance, maybe in future, I'll explain you that how this lidocaine or lamotrigine is playing a very significant role to understand this pathways of visual processing, right? So, okay. So here just these are some pictures, but due to the shortage of time, I'm not showing you or explaining you more about this picture. These are I wanted to show you what happens in this retinitis pigmentosa, how these rods and cones are getting degenerated. And there are few snapshots of this picture, which we captured in our lab. Okay, so again, I mean, this is the same picture of the slice of the retina. You can see the different layers of the cell, and the top you can see this is the photoreceptor layer, and this photoreceptor layer passes the information to the bipolar and horizontal cells. You can see here, and then they finally pass to ganglion cells, and from the ganglion cells, it will go to the optic nerve and to the optic chiasma, then to the brain, right, through the LGN. So this is how it, get processed. So you must be also, knowing, you must have heard from some newspaper or maybe some movies that many scientists around the world, they are talking about that can they restore sight by using some kind of bionic eye. So this is one kind of electronics chip they are talking about. You can see here, here I've written artificial silicon retina. Okay, so this is what they're trying to do, that those who have some problem in their retina, can we use some kind of electronics chip which can help them to see. And you'll be surprised. Here I'm showing you one picture, and the red color t-shirt guy who is standing behind this two person, he, the scientist, those who developed this bionic eye, and the person who is wearing this bionic eye, he is very excited that day because he is blind since birth. And he has not seen anything since the birth, and his wife is standing in front of him. Using this bionic eye, this person is telling that I'm so excited because first time in my life, I can see my wife. But the truth is something different. Actually, he is not able to see his wife. Only he can see some phosphines, some light and dark patches. So that means, at least, okay, whether it's a light or dark patches, but still he can see something, though not clearly his wife, right? So all this bionic eye, those who are working throughout the world, Harvard, MIT, Stanford, Berkeley, all this big universities in the world, they don't have the major success in this kind of this device. Why? Because the beauty is that we don't understand the brain and we don't understand the visual pathway, that how all this information get processed, as I shown you in this picture, that how all this visual information get processed in the eye level, and when it passes through the optic nerve and optic chiasma, which kind of information get processed, and finally, which are, being processed at the end in the brain. So this pathway is not understood properly. And if we don't understand all this mechanism, we cannot come up with the right kind of technology to restore sight to the blind, right? So that's why I'm very fortunate to work in a very interesting project. I'll highlight you here. So here, that's why my question is, will vision develops after surgery? Or if we can do some kind of treatment to the blinds, can we restore the sight in the brain or to the person? So here, I'm showing you this picture. You can see here, there is a very interesting hypothesis in medical science. It's called the critical period. Critical period hypothesis is one of the very fundamental hypotheses in medical science. And here you can see the two gentlemen, David Hubel and Torsten Wiesel, those who are the Nobel laureate. They got Nobel Prize in 1981, and at the time they are professor at Harvard University. And they gave very interesting hypothesis, which is known as the critical period hypothesis. So let me tell you, what does it mean? They told, actually they conducted many studies on the kittens, and from that results, they came up with the conclusion like this. Let's say I am born all right. Let's say today I took birth and I am completely fine, just like a normal baby, right? And just after my birth, you put me in a dark room. You keep me in a dark room. You kept me in a dark room, and in the dark room, you provided me food, drink, whatever I need, just like a normal kid, right? But I was not allowed to see anything. And I spent, let's say, 1 month or 2 months in the dark room, right? And after 2 months, if you'll bring me to the lighted part of this world, let's say that you brought me outside of the room. By that time, I won't be able to see anything. That means the neural pruning happens in the past few weeks or past few days or past few months in everybody's life, right? So that means as I was not exposed to the visual world just after my birth, up to, let's say, 2 months. That's why my visual neuron, the neuron responsible for the vision, they might have, or they are associated with some other job than the vision. That's why I want to be able to see. And this thing will happen to any kind of modality. Let's say if you are, if I'll, after the birth, if I'll put you in a soundproof room where you cannot hear anything, and if I'll follow this 2-month pattern, and if I'll bring to the this natural world, you want to be able to listen anything. That means the neural pruning happens in the past few days or few months of our life. And if you pass that period, maybe you will not able to get back that ability. Okay. And this is called the critical period. And these two scientists did many studies on the kitten, and they came up with this idea called the critical period. This is the time window is required in everybody's life to develop all the skill. So being a scientist, being an engineer, so we came across this kind of studies many years back, 15 years back. And then we thought, why not possible? Why the blind, if somebody is born blind, why they cannot see? And then we started one project because actually when we were coming across this kind of research, we found that all the studies being done on animal. But we asked ourselves the question, what about humans? Whatever we have done on the animal, we extrapolated this study on the human, but we don't know whether the human, will respond in the same way or different way. Again, in the same time, you must be thinking that all of a sudden, why you came up with this kind of question. So in engineering nowadays, if I'll ask you which are the buzzwords you are listening, definitely you will tell artificial intelligence. Artificial intelligence is a very big buzzword. Computer vision, robotics, these are all buzzwords in our engineering domain. So likewise, this also came to our mind that when we are making a machine to see, okay, basically, in the robotics, what we are doing, we are giving the kind of vision to a machine. But before that, we need to understand, let's say if I want to make my machine to see, before that, I need to understand as a human being, how I see this visual world, right? So this kind of question came to our mind, that do we understand how as a human being we see this world? If we can understand this mechanism, maybe we can develop the real kind of AI system which can do this job. And that time we came across different kind of studies, but we didn't find that anybody in this world had done this kind of research to understand how as a human being we see this world, right? So the question comes again, why the people have not done? Because in the world you will find millions of scientists, very big scientists. Why they could not answer this question? Because if you wish to learn, or if you wish to understand how we learn to see, then what you have to do? You have to do two things. First thing is, you need to do research on a newly born baby. Once the baby is born, and then from the first day, you will track how the brain is changing, what the information they can see. Most probably you can understand how we learn to see, right? But interestingly, baby, they don't understand what you are going to do, right? They will not understand your experiment. So that means you are not going to get the reliable data. First thing. Second thing is that the ethical board will not approve this study because you are not allowed to do any research on the newly born baby, right? That's why in the whole world, nobody has attempted to answer this question, right? So that time one question, came to our mind, that let's say, just imagine, let's say somebody born blind, has not seen anything, and now he's completely adult. Let's say he's 15 years or 20 years old, right? So now he's not a baby, 20 years old. And just imagine, by miracle, we restore sight to that person. Because in India, we know mantra and all this kind of thing, right? So we can just imagine that somebody who is 20 years old, has not seen anything since birth. Today, I did some kind of mantra and give sight back, right? And from tomorrow onwards, the person can see. And now he is 20 years old and he can understand what I'm going to do. So I can track their brain changes, or I can track how they're learning this visual world. And then this kind of knowledge will help me to develop the artificial intelligence agent. So I can give this kind of knowledge to my machine to see just like the human being, right? But when we came up with this kind of question, then definitely this critical period hypothesis was the main roadblock on our way. The critical period hypothesis says, no, it's not possible. If somebody has not seen anything since birth until 20 years, that means he is not able to see. And definitely when two big people though got Nobel Prize, and if you will challenge their hypothesis, definitely the whole world would be against you, right? And likewise, when we started this kind of work, the whole world was against us. Everybody was criticizing us that how come you can think that you can restore sight to the blind? But then we listened to our instinct. We listened to our inner things, that most probably we can make a headway in this domain. And we started a interesting project in India, which is humanitarian scientific project. Humanitarian means you must have heard about many research in the world where people do research, but there is no direct impact. But here, this is one of the most interesting projects in the world. Name is Project Prakash, and it is headquartered in Delhi. And now I am heading this charitable trust. And this is being founded by Professor Pavan Sinha, who is professor at MIT, and with whom I did all this kind of work. Okay. And what we do, this Project Prakash, this center is in Delhi, and we do outreach, treatment, research, education. That means we have a very big team. They used to go to different parts of India, UP, Haryana, Madhya Pradesh, Rajasthan, Himachal. From all these places, they screen the child, and they bring the child, those who are to whom we can restore sight. And then we provide the treatment, and then we do the research, that how just I mentioned to you, because there are many things we are still not, we don't know in the medical science. So through our research, we are providing many new concepts to the medical science. In the same time, this kind of research is helping us to advance the domain of artificial intelligence. And finally, we are providing education to this child, because those children, they have not been to school. And once they see, we think that we need to bring them to the mainstream society. That's why we are also educating them. We are providing them the education. So you can see here the doctor is providing the treatment. What we are doing, this is nothing but simple surgery we are doing. We are removing the lens, thick lens, which is called the cataract, and then we are putting intraocular lens, and then we start doing the research to understand how vision develops after sight onset. But the big question is, whether vision develops after sight onset. Let's say if we provide sight restoring surgery, according to the hypothesis, critical period hypothesis, can we tell that yes, the sight is, we are able to restore the sight? So we do many research. I mean, I'm not going to tell you all this kind of research what we do. We image the brain, we collect the brain signal, we do many computational modeling, and then we try to understand in the brain level, is the brain plastic enough to make use of information from the eyes later in life? That means whether the brain is still plastic, that means its ability to change again, to reorganize when somebody will start seeing this world. And in the behavior, can we see this kind of effect? So you can see here in this picture, I'm showing you two faces. One is 8 years old baby, another is 22 years old, one kind of adult, right? That means according to the critical period hypothesis, these two people have already passed that critical period, but whether they can see or not, right? And we do, as I mentioned to you, we do brain imaging. We track how the brain is changing, because I told you what we see, this is not the job of the eye, this is job of both eye and brain. And we do, okay. Just, I mean, I'm not going to talk very hi-fi things to you, definitely not, because you all are from different background. So just, I'm highlighting, I mean, in a very layman language, I'm telling you. This is one kind of study called the resting state functional connectivity. So basically, we image the brain. So you must be, you must be knowing that some people says that, oh, maybe he's intelligent because he's using his brain. Somebody is not intelligent, that means he's not using the brain. But the beauty of the biology is, biology does not sit idle. The neurons, they don't sit idle. They do their job, whether you're sleeping, whether you are awake, okay? So here, this study, I mean, what I'm going to show you one result that is called the resting state. That means when you are not doing any active task, I'll ask you to sleep inside this machine and I'll image your brain. And that time, I'll try to see how the brain formed the network. And this is called the resting state functional connectivity study. And you'll be surprised to know this resting state functional connectivity study is one of the most interesting research in the whole world right now. And again, the most surprising thing is that the scientist who came up with this idea that the brain can do many interesting things when it's in the resting state. This scientist is from Bhubaneswar. His name is Professor Bharat Biswal. When he came up with this idea, the whole world was against him, because how come you can tell this kind of thing is happening? But he is a very famous professor. He's in US, and he used to work with my lab at IIT. So this, I mean, okay, this is a very interesting kind of study I'm showing you here. So here you can see two brain picture. These are the brain picture. I'm showing you the left side picture. I'm showing you if you are looking the brain from the left side, from the left ear of your head, and the right picture is from the right side. Okay, this is the brain picture. And these are the activation of the brain. Okay, activation means, I mean, I'm just putting the layman terminology. So you can see the blue color and red color, right? So the blue color, that means if I'll take a signal out of this, wherever you see the blue, okay, if I'll take the signal, they will be highly correlated. And if I'll take a signal from a yellow zone, so that will be anti-correlated. That means you can see some blue activation in different parts of the brain, but functionally they are connected with each other. That means functionally they talk to each other, right? So this is a very interesting technique and very, I can say, powerful technique to tell many kind of things about the brain, whether the brain is having some problem or whether the brain is in the normal state. You can tell many things from this study. So I'm just highlighting one very interesting study. I mean, I'm not going into details. This is the picture I'm showing you. This is basically the fMRI images, and I collected the brain images, and this is what I'm showing you. This is the visual cortex. Visual cortex means back part of the brain. So you know this back part of the brain I'm showing you in my head. This back part of the brain is responsible for the vision, right? So I was interested to see how the back part of my brain is behaving when I restore sight to somebody, right? So here, T minus 2, you can see here, this is a 20 years old person. So, I mean, I've shown you in the picture. T minus 2 means when the person was completely blind, he has not seen anything. That time I took the images of the brain, and after analyzing this data, what I found a very big hotspot. You can see yellow color, very fire-like kind of hotspot, two places. I can see my cursor. Okay. T plus 7 means at T equal to 0 means time period 0 means we operated the person, we did the surgery. And after 7 days, Again, I image their brain. Then 30 means T plus 30 means after 30 days, and 120 means after 120 days. So you can see here, after 7 days of the surgery, this person, this hot spot, which is the fire kind of structure, big fire ball kind of, you can see, right? But after 30 days, the big structure is getting disintegrated. That means it's getting smaller or fragmented. But after 120 days, you can see there are many fragments, many small islands. So you can see T minus 2 or T plus 7, there is big island. And then along the timeline, after 120 days of the surgery, you can see there are many small islands, right? So what does it mean? Iska matlab kya hai, right? In information theory, I'm just putting one very engineering terminology to you. When you must be talking over your phone, you never, ask yourself how we can speak, how the information is going from my mouth to somebody's mouth. So this is, we call the decoding, how we encode or decode the information, right? So let's say, everything is digital. Digital means 0, 1, in 0, 1 form they work. So let's say I have 2 bit. Let's say I have 2 bit, right? And this bit system, I know. So if this is 2 bit and 2 bits are dependent on each other, that means this bit depends on this and this bit depends on this. And if I'll ask you how many ways they can encode information, so only 2 possible ways are possible because they are dependent on each other. That's why either they would be 0, 0 or 1, 1. Because they depend on each other. So just imagine if I'll make them independent, then how many ways they can encode information? So if I'll make them independent, they'll encode information in 4 different ways. That means 0, 0, 0, 1, 1, 0, and 1, 1, because now they're independent, right? So what we understood from this example, that when the bits are becoming independent, they are encoding more information. So this is the same thing happening in the brain. So here, all this hotspot, what you see, these are the neural firing, right? So initially, when the person is blind or after the 7 days of the blindness, the neural activation are either 1 or 0, 1 or 0. That's why you can see only big hotspot, right? But along the time, what you see, you can see many more island. In 120 days, you can see small fragmented picture, right? The hotspot. That means the neuron are getting more independent. That's why you are getting more different island. And when they're getting more independent means, scientifically you can say the brain is learning or the brain is reorganizing in its way. And this is one of the very interesting findings. So here I'm just showing you 6 subjects data that all of the brain, all of the blind children, after the surgery, they are also following the similar kind of pattern, right? And this is a very interesting kind of research. Do you know Charles Darwin? Charles Darwin, everybody know Charles Darwin. तो इनका great grandson का नाम है Horst Barlow, and Horst Barlow is professor now, currently the emeritus professor in Cambridge University, UK. So in 1980s around, he gave one hypothesis, theoretical hypothesis, that how brain encode information, but nobody could provide the justification for this kind of hypothesis. But first time in the world, we provided the hypothesis that how brain encode the information. And it is very much important to understand many interesting factors about the brain. So likewise, I'm not going to talk details here. So again, okay, here I'm just showing you development of functional specialization. What does it mean? You must have heard about another problem called the prosopagnosia. In prosopagnosia, the person, this is called the face blindness. The person is not able to see the faces. So the question is, why the person is not able to see the faces if somebody is having this kind of problem? So we are trying to understand which is the region of the face and how it develops, what's its origin. So this kind of study we do, okay? So I'm not going to explain you more about this kind of study, how the face area is developing. So from this, all this kind of research, what we found, the question which I asked a few minutes back, is the brain plastic enough to make use of information from the eyes later in life? So answer is yes. And can a child blind for several years since birth benefit from optical correction of the eyes? Then we found the answer is yes. So that means these 2 person you can see here, they have the ability to see. And from our research until now, we have restored sight to more than 700 blinds in India. You can see these are the snapshots of the blind children, those who were blind, but now they can see just like you and me. And not only they can see, but in the same time, They are providing us a unique way of understanding the brain and to answer many questions in the medical science. In the same time, the way they are learning this visual world, that is also helping us to develop many kinds of artificial intelligent applications, right? So, so I'm not going into details. So there are many interesting experiments we conduct, and based on that, we provide many interesting insights about this problem. So as I told you, that not only we restore the sight to the blinds, but in the same time we provide many groundbreaking hypotheses to the medical science, which was not there. And as I mentioned to you, please, I mean, okay, though I'm giving a talk, but one thing I can tell you here, always try to choose a problem which looks impossible, right? Which other people will criticize. And if you take that problem in a very scientific way, one day you will be the winner. The same thing I learned from my life. When I started this work, that time everybody criticized us. All the big people in the world, I'm talking, not talking about the India, about the world, they all criticize us. How come you can think that you can restore sight to the blind? But when we proved that yes, it's possible, now the whole world is following us. So this is the beauty of going in out of your way, right? So if you'll go in, if you'll follow the other people, then nobody might follow you because you are again one of them. But if you'll follow some different route, everybody will look at you because they, and main thing is that not only follow, but you have to prove it why you are different, right? So this kind of research is helping us to develop many kind of interesting AI applications, and which are being used in my lab at IIT Delhi. And also, I'm not going to explain you all this kind of, okay, there are many interesting research we are doing. So I just kept few slides, but due to the shortage of time, I might not be able to cover everything, but maybe in future, if I'll get a chance to interact with you, I'll cover many interesting things. And from our research, so one of the hypotheses, 300 years back, some scientist asked this one question, that how cross-modal ability develops in the human brain. Cross-modal ability means how, let's say, if I'm holding a pen, if I have closed my eye and I'm holding a pen, but still I can visualize it. Let's say if you're talking to your parents over phone, but still, though you can hear their voice, but still you can visualize them. This is the ability, one of the very interesting ability what we human being have. And this question was not answered properly. So we first answered this question in 2011. And that day when we answered this question, the cover page, and we published the paper in Nature, and then the cover page of New York Times covered our story. Finally, the Indian blinds could answer this question. So all of my research appeared in top-notch journal, like, you can see the Hindu covered my stories of one of my research. The Wall Street Journal, you can see. The Science magazine, the Science magazine is one of the top-notch scientific journal. They gave the cover page to our work. And you can see here, my student at IIT Delhi, they are working on a child, and we are recording the brain signal. We call the EEG, electroencephalogram, and my students are working on it. And this is one cover page of Germany magazine. So the Germany people, they visited my lab and covered this story, that what kind of research we do. So our research is appeared in many popular presses, Nature, Times Magazine, Harvard, MIT News, and many more. Even Netflix also did one documentary in my lab at IIT Delhi. You can see here they're recording different activity in my lab. So finally, I would like to say here, those who have the privilege to know have the duty to act. So we have just touched a tiny point of blindness in India, but our main objective is to make India blindness free. So now you know that there are many kind of blindness which you can cure, or which you can treat, or which you can help them to get back the sight. So now this is our duty to do it. So we cannot do alone, but if we'll come all together, maybe we can get rid of this big problem. You can see here, the child, when he came to us when he was blind, in the left picture, you can see the same child, and the same child who after the sight onset, when the child can see, you can see the smile in his face, right? And as a scientist, as an educationist, or as a researcher, this should be our objective. Always when we do our research, we need to ask ourselves whether we are going to contribute to our society or not, or how my research is helping the society. If you are not getting any answer, don't do this kind of research. Okay, so this is the trust I'm heading, and where we do many kind of, we do different kind of activity to get the donations, because actually these children, they are from very poor family, and we used to spend more than 2-3 lakhs for one case, and we used to take care of everything. So even sometimes we celebrate our birthday, and we spend lots of money, right? And even you'll be surprised to know many school around the world, the children, instead of celebrating their birthday, they give the money which are being used for the child education. Even you cannot imagine, even 500 rupees also matter. To provide a glass to a child cost 500 rupees, but you can also do in that way. So please join hand with us and make this world beautiful and makes India blindness free. And if you'll do good work, you can have a very broader impact on your society. So thank you very much. And I feel that the title of your FDP program is the right kind of program, because understanding fundamental science is truly the most important things to take a lead in not only in science, but also in technology development. Thank you very much. And if anybody is interested, actually in IIT Delhi, I organize one workshop. So the workshop I'm organizing here, brain mapping and artificial intelligence. If you are interested to understand more about the brain and what kind of information or what kind of things you can join from 11 to 19. So if you type brain mapping and artificial intelligence, IIT Delhi, you will get the link and you can register for it. Thank you very much for your attention. And if you have any questions, please, now, you can ask me.
Thank you very much, sir, for your informative session. Really, this topic is a heart-touching and a topic of social relevance, because giving vision to a blind, there is no other noble cause than this. And I really congratulate you for taking up such a nice work, which is fruitful to the society, because a lot of research is done, but ultimately the benefit to the society is more important. And I really congratulate, I appreciate your efforts for giving vision to the blind. And really it's remarkable. It is not possible to express in terms of words, but a lot of after hearing to your lecture, a lot of respect for you came to my heart, and also all the participants. Thank you very much, sir. So now the session is open for question and answer. I request the participants to ask some questions.
No
Dr.
question.
Gautam.
Dr. Tapan, actually, I want to know, suppose a child is born is blind. So how can we know that? That means all or all blind child may not be able to see. So how can we know that? That means which blind child has the ability to see?
Hurricane. Okay, very good question. So there are different techniques are there. So basically, your question is, as I mentioned to you, that there are different kind of blindness, and all of the blindness cannot be cured until now. But as I mentioned again, that nearly 40% of the blindness can be cured. So we are working on that 40% blindness. So the main question is, let's say, okay, as I told you, eye and brain, they're the two important components. So first you have to check whether eye is absolutely fine or not. You have to check whether there is a proper connection from the eye to brain is there or not. If it's not there, then also you cannot do the things. So now, starting from this kind of blindness, as we know, just like I gave the example of cataract. Cataract means when due to this viral infection or due to this any other kind of infection, if that the layer, the opaque, the layer is developed, the lens get opaque, you cannot see anything. But in that case, the eye is connected to the brain. So we do certain kind of test by which we can access that, yes, the blind can see. At least, if they have the sense of light and dark. Let's say if I'll switch on the light and dark, and if I'll ask somebody, can you feel it? If they can feel it, then I know that, yes, there is the link between eye and brain. And most probably we can rectify it. So this is how we do. But working on this kind of patient also, they are also helping us to understand which part of the neural pathway or the visual pathway is doing what kind of job. So once, I mean, okay, this is one kind of top-down or you can say bottom-up process. So as we understand this kind of problem, then maybe we can move forward to address the other kind of blindness. I hope I gave your answer.
Thank you.
So any more question from the participants?
I think your participants either understood everything or they could not follow my talk.
No, no. So thank you, Dr. Tapan Kumar Gandhi, sir, for sharing your valuable knowledge and research work with all the participants. And please accept this e-certificate on behalf of Roland Institute of Pharmaceutical Sciences as a token of appreciation.
Okay.
Thank you very much, sir.
So it's done. Can I take a leave? I have to join
Yes,
another.
sir. Yes. Okay. Okay. Thank you very much, sir.
Thank you, Dr. Nivedan, for this opportunity to deliver a talk in your program, and definitely Dr. Gautam.
Thank you, sir. Thank you.
Okay. Okay.
So, dear participants, we have another lecture at 2:30 p.m. So, all are requested to please join 5 minutes before the session.
ମାଇକ୍ରୋ ସେଠି କହୁଛି ନାଇଁ
हाँ
ଫାଟିଥିଲା ସୋଲାର ଟେବୁଲ ଲଗାଇଥିଲା ଯେ ଏବେ ଗୋଟେ ପିଲା ଲଗାଇଲା ପୁରା
हाँ हाँ एटा कौन करियो पड़ियो
ଫାଇବର ଦୋକାନରୁ ପଚାର
हाँ फाइबर वेल्डिंग कर देले हो पार्टिसिपेंट्स ये देखिए पड़ियो नहाती बेसी
Hello, am I audible? Hello.
Anup sir, can you hear me?
Yes.
Okay.
Hi sir, how are you, Ravi sir?
Fine, Anup. How are you?
Yeah, I'm good.
Thank you for accepting our invitation.
Thank you. Thank you for inviting.
And welcome to RIPS virtually.
Yeah.
आप कैसे हैं?
Yeah, I'm good. So, how is life?
Going on?
Yeah. Dr. Niranjan, I just wanted to just
Yes.
help me in actually projecting my screen. Like, I'm just trying to see the options.
Yeah, there is an option called as share.
Here, yes. I think you are currently you are sharing. That's why
I
it is.
have shared. I will stop that share and then you can share. There is no problem.
No,
And
no
in
issues. No.
And in your screen there is option more.
Yes.
It is written. If you click at that, you can rename also.
Okay.
There is option for rename.
Where it is?
Just
You go to participants list is there, na?
Participants.
Yes, participant is there.
Right hand side it will come list of participants.
Yeah.
Top one will be your name.
Correct. Can I do that change? I'm just checking. If you can
Click on the
make this.
video.
Okay.
Camera.
I have to click on the video.
Means right side to your name. Video icon is there, na? Camera icon. Click there.
Okay. Not very conversant to Zoom. Actually, we are using day in day out Teams, Microsoft Teams.
Okay.
Yeah. And most conversant to Teams as well as more conversant to also Google Classrooms.
Okay. Teams is very costly.
Yeah, because it's a like our organization has the Outlook. That's why they give Teams. Somehow I'm not able. Yeah, I'm in home today. I'm in home.
Okay. So you got the icon to change your name?
Yeah, I am doing that.
Okay.
Yes, I did that.
Yes.
Sir, Narendra sir, shall we start?
Yes.
Sure.
So thank you, Mr. Anup Abhijit Chaudhary, for accepting our invitation. I request the co-coordinator for this program, Mr. Kanu Charan Panigrahi, to introduce our resource person to the participants.
Good afternoon, one and all. It's a great honor to introduce Mr. Anup Abhijit Chaudhary, who is working as a global head of global topical and differentiation formulation in Dr. Reddy's Lab Limited. He is seasoned pharmaceutical product development professional, having 18 years of experience across state-of-the-art R&D organizations like Dr. Reddy Limited and Renvex Limited. Currently heading the global topical and differentiation formulation R&D at Dr. Reddy Limited. Experience in end-to-end product development from conceptualization to commercial production of various solid, oral, and topical dosage forms for global marketing. Is proficient in development of differential formulation from preclinical through the late stage development and phase 1, 2, 3, 4 clinical studies. He is currently leading a team of 35 highly skilled formulators. Among them, 10 are PhD holders and 25 are M.Com candidates. He has contributed in co-inventor in number of international patents. He has co-authored number of research articles in various national and international journals. He contributed as co-author to book chapters for reputed publishing house. He represented currently the organization various international and national conferences and delivered seminars and presented posters. He mentored many graduates, postgraduates, and PhD students from premier educational institutes like NIPA, BITS, IIT, BHU, etc. He started his professional career in 2003 and worked as junior manager in Dr. Reddy, Hyderabad. After that, he joined in 2006 to 2006 in Ranbaxy and worked up to 2011 as senior research scientist. Presently, he is the head of global topical and differentiated formulation in Dr. Reddy Limited, Hyderabad. He done his M.Pharm, B.Pharm in Sri Jaydev College of Pharmacy in 2001. He completed his M.Pharm in 2003 and done his MBA Management Development Program from IIM Bangalore in 2016. With this, I welcome you, sir, to address the gathering. Thank you, sir.
So, Anup sir, please unmute yourself.
Okay, is it visible?
Yes,
is it audible now?
Yes, clearly audible. You can share your screen.
Yeah, so I'm doing that. I'm just trying to see that.
Share screen.
Yeah,
share screen. Then screen.
I can do the better. I can directly go to the presentation.
Okay.
Is it visible?
Started screen sharing. Just wait. Now it's coming. Yes, visible.
Okay. So first of all, thank you very much, Dr. Niranjan, for inviting me to this seminar, and Dr. Kanu for a nice introduction, and Dr. Ravi for extending his invitation and warm welcome to this seminar. So, friends, I'm highly delighted to share my thoughts and my experience with respect to differentiated formulation to all of you. Differentiated formulation is very close to my heart, not only because I work on this subject and I'm currently working and I'm still practicing since last 18 years. But the differentiated formulation, what it makes different from other dosage forms or other product development is majorly it is very much patient centric. And when we are developing differentiated formulation, it is completely the patient is always at the center, and we it is kind of like a if we are not actually working against the disease, we are actually working for the patient. And when we are trying to kind of like develop anything with respect to a differentiated formulation, we first see that what is the unmet need. And I will just this complete session, I don't want to be a monotonous, and rather actually I'll be more than happy to be stopped anytime, and any queries, any clarification is required, then and there itself you can kind of like clarify with me. And if you have any questions, queries, please do not hesitate to stop me. And rather actually as a monotonous talk, I want to have an interactive session so that I'll be able to understand whether you are able to actually follow what I'm trying to tell. And if anything more needs to be emphasized, I'll be able to kind of like give a little bit importance to that and go a little bit deeper into that. So in today's talk, actually, we will cover. So I just wanted to actually introduce all of you towards what is differentiated formulation is about, and how it is important, and how it is actually different from other product development. Second thing, I will try to give a perspective of the unmet needs and how we actually try to come up with different differentiated formulation proposals. And I will take you through some of the examples or the case studies where you will be able to understand more that why it is differentiated formulation and how it is different from other drug development. hope to have a very interactive session. And please, as I told, please stop me, or please raise any questions if you feel needs to be answered, and don't wait for anything. Just stop me and I can give you that answer. Let me just open the pointer so that I'll be able to easily take you through. So to start with, actually, I just wanted to give you a perspective of the whole pharmaceutical product development. So this is a kind of like, I wanted to actually show you a graph, or a 2x2 graph, where I just wanted to show you that how different kind of product development categorizes with respect to the rewards and investment or risk. And this risk, the x-axis, so it is the risk is actually the risk of failure. So how it is actually segregated with respect to product development. As you are aware that we are working for a commercial organization, and whenever we develop a formulation, obviously the first thing is that we want to come up with a solution for the patient. But at the same time, the pharmaceutical industry, to be viable, it needs revenues as well. So that's why this you can see that different pharmaceutical companies has segregated into different categories. So to start with, so if you see, the NC comes to the extreme right of the thing, where the reward is very high. When I tell it is reward, that if you have an NC, it is like a new chemical entity. So it's a it can go into a multi-billion-dollar kind of like revenues. At the same time, it has a lot of risk with respect to the investment, because for developing a NC, at least you need, at this moment of time, minimum $1 billion is required. And I was recently going through some literatures which says the current average of investment for developing a NCE has gone up to around 2.5 billion, and 2.5 billion is not a small amount. And that's how you can see many Indian companies are not into this kind of business, because here the investment risks are high, and the risk of failure is also more. I am sure that you people are aware that NC is new chemical entity, and it has to be start from the scratch. And when you are developing a product from the scratch, you have to develop a molecule, work on the medicinal chemistry, work on the kind of like the animal studies, and all together you have to take the product from the phase 1 to phase 2 to phase 3, and also phase 4, the post approval surveillances we have to do. And where the risk of failure is very high, because 1 in a 10,000 molecule gets actually gets a chance to go to the market, or it gets marketed. And once it is marketed also, there is no guarantee that your product is going to be multi-billion dollar success, because it all depends how the product is positioned, what is the market, what is the kind of like patient pool, and what is the price point we are able to put. And always it has a risk of actually the generics coming in and devaluating your NCs. So that's why many Indian companies are not into NC business. Then comes to the extreme left corner and extreme bottom corner are the generics. And generics we know very well. Many Indian companies are actually working on this front. Generics are the kind of like the bioequivalent or the therapeutic equivalent product of NCs or the innovator products. So it's a very crowded market. That's why today, if you see any product which is getting actually the patent is getting expired, you can see that more than 20-30 players are there in the market for generic. And once the product is getting genericized, the price of the drug is actually kind of like deteriorating like anything. And I have seen that sometimes the price has actually deteriorated to kind of like 1/100th of the price of the innovator. Like for example, if your innovator is actually selling in 1,000 rupees one product, when it is genericized, I have seen it has gone to 10 rupees also. So in that case, although there is a still profit to the organization, but the reward in terms of actually the complete kitty is nowhere actually similar to the NCs. It is very low, but at the same time, how it is beneficial, it has a very low risk, because now many organizations know how to develop generic products, and that's how many people are coming into this. And generics are also very widespread. If you see, generics are having low complex, medium, moderate complex, and high complex. So high complex generics people are very low, but if you see low kind of the low complex or moderate complex generics players are many. And it doesn't take much time with respect to developing a product. It takes around 2 to 3 years to get a product approved from US perspective. And if you see the rewards also, sometimes you get sometimes you get moderate rewards. So it may not be very high with respect to in comparison to NC, but still it is very decent. That's why if you see India, many companies, many pharmaceutical organizations are actually pitching into this generic business, where the rewards are low, but risk is very low. Then comes to the differentiated formulation, where it is actually a in between with respect to the NC and generics. So differentiated formulation, how it is different, I'll tell you that. First thing is, what is differentiated formulation? So differentiated formulations are always the old molecules. So these are not new chemical entities. These are always new old molecules, but in a different avatar. So what it is actually, it is kind of like a the current gold standard products. What is the lacuna in those things, or what is the demerits in those things? How it is actually not helping the patient? We try to understand, and that how it is not helping the patient is called unmet need of the patient. So this differentiated formulation is always designed around the unmet need of the patient, and that's how you can see it has a moderate risk of failure. At the same time, it doesn't need as great as investment like NC, but it is a little bit higher comparison to generics. If you see, rewards are also moderate, because you are working on a space where many players are not there. Because when you develop a differentiated formulation, you are kind of like the first to develop this kind of formulation, and that's why many players will never be there in this kind of like sphere. And if you see, even the biosimilars or the biologics also kind of like share the same space. But today, for the interest of this topic, we will not discuss more about the biosimilars. We'll more focus on the differentiated formulations. And as I told you, the golden word of differentiated formulation is unmet need. I wish to take you through a little bit more with respect to the unmet need as well. So if you see, the unmet need can be anything. So unmet need can be a unwanted adverse event. Like for example, you know that many NSAIDs, the painkillers, so they have a common side effect of gastric side effects like ulceration and other kind of like gastric gastrointestinal issues. Then if you see, there is a problem with respect to lack of efficacy. Like for example, if you are in actually pain, and anybody won't if somebody wants to give a medicine which is going to work after 2 hours. So as a NSAID or as a painkiller, the patient needs that immediately it should work. So as much as less time, it is good for the patient. Like somebody is vomiting, and we cannot give a product to him or her that which will work after 3 hours. or kind of like if somebody is actually having kind of like vertigo or sneezing or kind of like mainly rhinitis, and if you see this kind of like the skin diseases with like for example insect bite. So you don't want actually the medicine should or the drug should work after a long time. It is always good that it should be working as fast as possible. So this is something kind of like a lack of efficacy. Or sometimes we see that you need to take a lot of pills to get that efficacy. Like for example, there are many drugs which are having very low viability. Even there are drugs which are having less than 10% viability. So most of the time actually the dose will be very high. Sometimes the patient has to take multiple pills, and there it comes the lack of patient comfort or convenience. Like there are IR products, immediate release products, which kind of like is given at least 2 to 3 times a day. And if you see the disease is a chronic in nature, and if it is kind of like has to be taken for a longer period of time, how patient inconvenience or discomfort you can understand if you have to take a medicine 3 times a day for next 3 months. So this is kind of like a unmet need where the lack of patient or comfort or convenience comes. Then comes the lack of dosing compliance. Like for example, there are a lot of medications which tells you that it has to be taken empty stomach, or with food, during the food, or kind of like it has to be taken only in the night time, or it has to be taken only in the daytime. Then it gives a kind of like a dosing compliance. Like the patient will not be able to adhere to the dosing compliance many times, because sometimes the patient is actually kind of like traveling, and he or she doesn't have that kind of like he or she is busy in something, and he forget to take the medicine as per the dosing recommendation. So is there anything we can do for that? So that is kind of an unmet need for this. So another thing is that uncompromised or the compromised patient quality of life. For example, you are taking a product, a drug for antibiotic. The general antibiotic, if you see, the major side effect associated with antibiotics are diarrhea. And for example, any somebody who is actually working in an office or doing a business, and you tell that every day you have to go and take an injection. Is it comfortable for a patient? No, it is not. And if the dosing or for example, many anti-cancer molecules, these are coming in as injections. And most of the cases, like when we give the anti-cancer drugs, those are IV infusions, and it has to be given under supervision of a medical practitioner. And every time the patient has to be admitted into a hospital, and somebody has to actually kind of like go and understand whether it is like and you can understand like how much discomfort for a person who has to go for every day or every alternate day to hospital to just take a injection or get kind of like admitted into hospital. So these are certain things like unmet need. I want to also spend 1 or 2 minutes over here that how we can actually avoid this kind of things. Like let us consider that I was telling about the NSAIDs having gastric irritations or something. Can we come up with a formulation which is a combination which can take care of the side effect? So that can be that can avoid this adverse effect. That's why you could see that many of the formulations, like nowadays it is coming up with kind of like mostly the NSAIDs are given along with PPIs, the proton pump inhibitors, or the gastrointestinal or the gastric motility drugs. So those kind of things we can come with a combination, or like we can come up with a combination with probiotics, so that the adverse reactions or adverse events can be reduced. Similarly, if you see lack of efficacy, can we come up with a formulation intervention so that the viability of the overall product can be improved, so that the pill burden can be reduced, or sometimes like if it is a NSID, and if it is kind of like having a very kind of like longer Tmax, and it takes kind of like 2 hours to onset the action, or 1 hour to onset the action. Can I do something with respect to formulation interventions that the products start working in 5 to 10 minutes? Like lack of comfort or discomfort, what patient feels like a product is having 2 to 3 times a day and he has to take for 30 days. Can I make a formulation which at least can be done, at least can be given once daily? So IR to ER, IR to CR kind of formulation I can make. Then lack of dosing compliance. For example, something has to be taken without like kind of like before food or after food. Can I make a formulation intervention so that I can claim it can be used without regards to food? So irrespective of your food habit, it's irrespective of your dosing before food or after food, the efficacy is going to be same. If there are ways to do that. And like compromised patient quality of, like for example, I was telling about the hospitalization or injectables. So can I convert these injectables to solid orals, where you don't have to actually wait for the any medical practitioner to go and or hospitalized to get the medication? You can have those medications very well. See, I can give you examples like there are some anti-epilepsy drugs where just before 2 years there were only injectable formulations were available. And imagine a case, somebody is like is suffering for epilepsy and gets seizure. Do you feel that he or she will be in a condition to go to the hospital or go to a medical practitioner to get rid of that? But now we have actually come up with formulations which we can give to the patient which are already diagnosed with epilepsy, so they can actually maintain that therapy in home. And in case somebody actually still it gets actually kind of like somebody gets seizure, if somebody is there in home and they can actually, it is a solid oral tablet or nasal spray, anybody can actually just administer the drug to the patient and the patient can actually be saved before nobody like the patient is not required to be taken to the hospital or the medical practitioner immediately, and we can save a lot of life through that. So I'm just trying to go to the next. So these are something which are very much within the scope of differentiation. So as I told just before, like the reduction of the pill burden. So when you are actually taking multiple drugs, like we are taking 3 different drugs which can be kind of like combined to a FDC, so that the pill burden can be reduced. There are products which are having high dose but having less viability. Can we reduce that? Can you increase the viability so that we can reduce the dose, so that we can kind of like reduce the pill burden? There are products which are to be taken 3 to 4 times a day. So can we make it to a OD, once daily formulation, so that we can reduce the pill burden? So then change in route of delivery. Like for example, there are products which when it is taken orally, it has a lot of side effects, like NSIDs. When you are taking NSIDs, there are a lot of systemic side effects. But instead of using oral or taking the medicine orally, we can make a formulation topical. So the route of the kind of like delivery can be changed to topical, so the systemic side effects can be reduced and the local effect can be maximized. And if you see the kind of like reduction in dosing frequency, I just told like when the product is given 3 to 4 times, there are many ways to reduce the dosing frequency by making it to a controlled release or sustained release formulation or delayed release formulation. And also most of the time actually there are old drugs, but we try to understand from the mechanism of action or their side effects whether it can be repurposed for any new indications. Like I can give you some examples. Today also I'm going to cover many of them where you can see that repurposing has been done. Like maybe a drug which is having NSAID property, but can be what we have seen is that it can be also repurposed for rheumatism. It can be repurposed for any other diseases. So we are going to kind of come up with this kind of examples. And formulation modification, if you see, most of the time we try to, when I'm trying to tell it is a formulation modification, it is mainly with respect to the modification of the PK parameter. Like if I can come up with a formulation which can be the onset of action can be improved, so that can be also a formulation modification. So before I go to the case studies, so I want to actually, next is actually I have some case studies. I wanted to take you through those case studies. I just want to take a pause and understand if somebody wants a little bit more clarification on this.
You can go ahead with your presentation. Okay. If
Yeah.
any question comes, they will type in the chat box, and I will forward
Got
that to you.
Got it. Oh, Dr. Nandan, just check. Am I going very fast? Do you want me to?
It's absolutely fine.
Okay. Thank you. So the first case study is actually, if you see, it's a tofacitinib citrate molecule, which is a Janus kinase inhibitor. And initially, when Pfizer came up with this product, it is actually for the management of rheumatoid arthritis. And the dosing is 5 mg, which is given generally for twice a day. And if you see how the Janus kinase inhibitors like tofacitinib, how it is works, you see the majorly it is actually down regulates the cytokines production, and that's how you are aware that cytokines are majorly responsible for the rheumatoid arthritis, and it is actually down regulating the production, and that's how actually it is acting on the rheumatoid arthritis. And as you are aware that rheumatoid arthritis is a disease which is chronic in nature, and generally the treatment regimen kind of it spans from 2 to 3 months to sometimes it goes beyond 3 months as well. And if you see what Pfizer has actually done is that initially they come up with the IR formulation 5 mg, and then they understood that it is a chronic disease, and it has been taken it is actually recommended to be taken twice daily, and it has to be taken for a longer period of time. So they understood this unmet need, and immediately what they did is actually they came up with a formulation, XR formulation, and that's how they have improved on the dosing compliance, and that's how the better patient compliance is accepted and achieved, and life cycle management is also for the product. See, for the differentiated product, although it is completely aimed with respect to the patient, but at the same time the commercial organization also gets benefit out of that. See, they were coming up with they are able to come up with a formulation which can actually kind of like a new or the life cycle management of the old drug and old product, and now they are continue getting the revenues. So I just wanted to give you a little bit more with respect to this how they have developed this product. So if you see, so initially they have come up with 5 mg IR, and which has to be taken BID, and they proposed a 11 mg of XR formulation, which is a once daily formulation. And I have told that it is a rheumatoid, it is indicated for rheumatoid arthritis, and the dosing period, the treatment period is relatively high. And it always poses a difficulty for a patient who has to take BID for a longer period of time. And when you are giving IR formulation, there is always a chance of actually fluctuation of your the concentration of the drug in the blood, because IR formulation acts as actually there is no control on the release. Actually, so all the drug is dumped, it gets absorbed, it gives you Cmax, and kind of like then it starts actually reducing as the drugs gets eliminated from the body. So that's
Excuse me,
how.
sir. Excuse
Yes.
me, Arnab, sir. There is a question from one of the participants, Mr.
Yeah.
Romeo Bhushan Mishra.
Yeah.
What he has asked is, are novel drug delivery dosage forms and differentiated formulations similar, or what is the basic difference between both?
Okay. So I will tell you that differentiated formulation utilizes novel drug delivery systems, but the reverse is not true. So when it is a differentiated formulation, it may not be novel also. Like for example, I'll tell you, you are aware of the kind of like the Alzheimer's disease, or let us consider that Parkinsonism. Okay, when a patient actually undergoes Parkinsonism, as you know that they're actually kind of like they get a body shake, hand shake, and they are not in a position certain time that they can even kind of like hold a tablet and kind of like administered themselves. So a small change in the tablet design, so that the patient is able to pick up the tablet and hold it well and able to administer, also can be a differentiated formulation. But this doesn't utilizes any novel drug delivery system. It is just a design of the tooling. So you are able to come up with a formulation which is helping the current patient unmet need. Hope I am clear.
Yes, sir. Yes, yes.
Thank you. So, so if you see, I was on how the differentiation Pfizer has brought is actually they have actually come up with a formulation, XR formulation, the extended release formulation, which has replaced the 5 mg BID with 11 mg QID. So in the right side picture, you can right side graph, you can see these are the kind of like the predicted steady state PK profile when you are giving the 5 mg BID formulation, IR formulation. And when Pfizer actually predicted the PK of the MR formulation, what they targeted is, see, the IR formulation is able to actually take care of the MEC values up to 6 hours when it is given once. So as the dosing recommends 2 times a day, so altogether 6 hours on 6 hours, so 12 hours dosing regimen is able to kind of like it is above the MEC. And what they developed, so they developed a XR formulation of 11 mg, and you see, so they are able to now come up with a once daily formulation which is able to take care of 12 to 14 hours above MEC. So that's how they have come up with a formulation technology called osmotic drug delivery system. You people are aware of what is osmotic drug delivery system, where it is and there are many types of osmotic drug delivery system. This is a very preliminary type of osmotic system which is called elementary pumps, and where the drug is actually kind of like the drug will be there in the core, and it will be having some osmogens, and it will be coated with a semi-permeable membrane, and there will be a laser drill hole. And when a patient takes up the medication, the GI fluids enters through the semi-permeable membrane, and that's how the osmogen helps in kind of like absorbing the GI fluid into the formulation, and that's why a pressure is created inside, and that's how the because there is a hole in the laser drill hole in the system, and the drugs escapes out of the formulation in a very controlled manner. And I'm sure that you are aware that generally osmotic drug delivery systems gives zero order release, because they are able to, irrespective of the kind of like the condition, they are able to release constantly through the dosage form, because it is regulated through a osmogen, and the drug is coming through a laser drill hole. And why they have actually gone for osmotic drug release system? Dr. Niranjan, you have a question?
No, sir. Please go ahead.
Thank you. So, this why they have gone ahead with this laser drilling technology, or is I'm sure that you are aware of what is the difference with respect to release kinetic of osmotic drug delivery system, or in comparison to any carrier mediated drug delivery system, or the kind of like, a reservoir based drug delivery system. So osmotic is also a reservoir drug delivery system, but in contrary to other reservoir based drug delivery system, it constantly gives equal amount or constant drug release, irrespective of the condition, irrespective of the pH condition, irrespective of any external factors it gives. And the beauty of osmotic pump is the product or the drug comes out of the formulation as a suspension, whereas in most of the reservoir based system, the gastric fluid has to penetrate into the semipermeable membrane, the drug has to be dissolved, then only it comes out of the formulation. That's why tofacitinib is a kind of like a pH dependent, having pH dependent solubility. It has a very high solubility at lower pH. So it has a high solubility in the stomach condition, but when it goes to the lower intestines, it has a lower solubility. And at that moment of time, when the GI fluid enters into the formulation, it will not be able to dissolve. And that's why the Pfizer very carefully developed this product and as a osmotic drug delivery system, so that the when the drug goes through the GI tract, irrespective of whether it is a lower pH or higher pH condition, like the stomach to jejunum, ileum, rectum, it is not dependent on the pH of the gastric fluid. It only needs the gastric fluid, and once it is actually the GI fluid is there in the formulation, it constantly pushes the drug out of the system as a suspension. And as it is coming in very low quantity or very continuous and constant manner, it is able to kind of like give a sustained release or a controlled release and a zero order reaction. And because it is coming in a very low concentration, the available GI fluid is able to dissolve it and take care of the absorption. And Pfizer has actually kind of like given a name to this system as extrudable core technology. Although it is a osmotic drug delivery system, they have done it's kind of an elementary pump, and that's how they have actually coined a name extrudable core technology. I would like to also take you through some of the pharmacokinetic data. So if you see, when they have done a study against the 5 mg twice daily. So if you see, this is the first dose of the 5 mg, this is the second dose of the 5 mg, and they have compared against the 11 mg XR. And you can see the ratio. This is the study where it is 11 mg versus 10 mg, like 5 mg BID. This is day one study. So day one study, you see the adjusted ratio of the T/R. If you see for AUC, Cmax, everything is well within the kind of like well within the criteria of the bioequivalence. At the same time, when they have actually seen what happens to the steady state, so they have done multiple dose study, which is a day 5 study. So on the day 7, the fifth, when it is like after fifth multiple dose, they have checked on day 7, and you can see this is the 5 mg BID dosing, and this is the 11 mg dosing, and you can see both Cmax and AUC are well within the criteria of the bioequivalence 80 to 125. These are the C trough and C mean. So these are, as you know, that this is a controlled release formulation. It is supposed to come lower than the IR, and it has come, and it is rather, it is good, because if you see in comparison to IR, the C trough and C mean are very low, and it will be always good for a product which is actually to be given kind of like a for a longer period of time, so that the exposure, the overall exposure is not compromised. However, the highest concentration is moderated in a way so that you can reduce the side effect. So this is how actually they developed a formulation which is to be given BID to OD. And also what they have done is that they have tried to understand whether there is a food effect in their formulation. So now you see they have given their XR formulation fed and fasted, and you can see only the Cmax is different, and that too it is just kind of like shooting off little bit on the border. But if you can see, the AUC is almost similar for both fasting and fed and Cmax, and that's how they have come up with a formulation which can be given irrespective or without regards to food. Hope this is clear. So just if you have any question, please feel free to ask. So then we will go to the case study 2, where I'm just taking the same example of tofacitinib. Pfizer came up with the drug first for the rheumatoid arthritis, and subsequently they started looking into the different mechanism action of JAK inhibitor, and they understood that the JAK inhibitors, the Janus kinase inhibitors can also work against the ulcerative colitis. And they came up with a formulation which is, and only thing is that initially when they found out tofacitinib, or they discovered tofacitinib, they were thinking that it will only work on the rheumatoid arthritis, and the dose is 5 mg twice a day. But when they discovered that it can also work on ulcerative colitis, they have actually seen that it is working at a higher dose, which is 10 mg, which is to be given 2 times a day. And initially they again came up with 10 mg BID formulation, and similar story goes. After that they came up with 22 mg XR formulation, which again give improved dosing compliance, better patient compliance, and life cycle management. So just I will not take much time, because I have already covered that. You can see the ulcerative colitis, where the drug was to be given like 10 mg BID. For IR formulation, now it is 22 mg ID, and you see the general kind of like dosing recommendation is actually your 8 weeks, and which generally extend to 16 weeks. So you can see that 2 to 4 months the dosing period is, and that's how you can see there is a lot of patient comfort when it is given once a day for a longer period of time. And you can see that initial phase, like the maintenance phase, how can be, it can be initial phase, it is given like so this is the induction phase, and the maintenance phase can be given like 5 mg BID or 11 mg OD, or 10 mg BID, 22 mg OD. So depending on the disease progression and the disease condition and the severity of the disease, it can be modulated. So they have 11 mg XR, 22 mg XR. So they have given a scope to the medical practitioners that they can actually dose kind of like they titrate the dose. They can start with 11 mg and go to 22 mg. So depending on the nature of the disease or the advancement of the disease. And this is how I told, like how they have come up with 5 mg IR to 11 mg XR. Similarly, they have come up with 10 mg IR to 22 mg XR. And I just wanted to also give you some perspective with respect to how they have evaluated their 22 mg XR with respect to IR formulation in the pharmacokinetically. So you can see this is 22 mg versus 10 mg BID. And for AUC, because ulcerative colitis, Cmax is not much important, and when you are kind of like formulating a doses from IR to XR, there is always a possibility that your Cmax will be blunted, so that for when you are converting from IR to XR, the regulatory requirement is also not that you have to match with respect to Cmax. Generally, for chronic diseases, most of the time it is actually the AUC is more important in comparison to Cmax. That's how you see ulcerative colitis is a chronic in nature, and they have just tried to understand how it is spanning with respect to AUC infinity and AUC last. And you can see that the 10 mg IR versus 10 mg IR twice a day versus 22 mg XR is dot 100%. So it can be easily replaceable, and now it comes with the patient compliance as well. Similarly, if you see, this is kind of like a faster study, and this is a food effect. So the 22 mg, they have seen that how their product behaves with respect to the food. So the 22 mg XR itself has been given as fasted and fed condition, and you can see it gives further comfort to the patient that he or she don't have to actually kind of like take this product to the dosing regimen, like as per the specified dosing condition. It can be taken irrespective of the food, and that's how it gives further compliance to the patient. So these are two examples where you could see that there is a improvement in the patient compliance because your dosing frequency has been reduced, and from how the pharmaceutical organization comes with initially comes with one kind of like disease recommendation or indication, but eventually they are foraying into different kind of like clinical trial and understanding the behavior of the molecule, and from the mechanism of action and their side effects, they're understanding where other places it can be also used, and they are repurposing that medication for the different other indications.
Excuse me, sir, I have a question.
Yeah.
So when in this 10 mg IR, this AUC is for 2 tablets.
Yes, it is for 2 tablets.
Okay.
Because
So
it is BID. So twice daily you have to give. So 10 mg has to be given twice daily, and 22 mg as a MR formulation or XR formulation, it is given only once a day.
So I have one more question. Generally, whatever we have studied in our books, it is recommended that those drugs which are having by half-life between 2 to 6 hours are suitable for sustained release formulation.
Yes.
But what we are seeing in the current scenario of industries, irrespective of the half-life of the drug, they are developing sustained release formulations. Can you throw some light on this aspect?
Sure. So you are absolutely correct with respect to the fact you have told. If you see in general, the candidates which are having 2 to 4 hours or 1 to 4 hours as a kind of like half-life, they are kind of like very suitable candidate for making a extended release formulation. However, those are kind of like bit limited, where only the like when somebody wants to only come up with a formulation which can be replacing the IR to XR with respect to only the number of doses, like you want to actually reduce the number of doses to 2 times to dosing regimen to only 1 time, then what you are telling is correct. But when it comes to the safety of the molecule, when it comes to kind of like efficacy of the molecule, most of the cases actually we are developing formulations where the therapeutic window, when the therapeutic window is very narrow, it has to be developed as also kind of like a sustained release or controlled release formulation. And when actually we are developing this kind of formulation, we ignore that whether it has the Tmax between 2 to 4 hours or not. And sometimes the dose has a kind of like it has a safety with respect to dose. Like for example, the recommended dose, like for example, there is a product called Aqis. So Aqis is currently given 100 mg 3 hours a day. But what you have seen is that if you give multiple doses at a time also, there is no issue. So what we are trying to do, and it has a very high T-hub. Like for example, it takes it is more than 10 hours. 10 hours is the T-hub, but still what people are actually coming up is that they are coming up with the extended release formulation because it has a high tolerance, and the maximum the toxic levels are very high. So hope
Yes,
I
yes.
could make you understood like
Yes.
why we are coming up with formulation, extended release formulation, irrespective if the Tmax is lower or higher, or it is not fitting into the criteria of general criteria of 2 to 4 hours.
Thank you. Thank you for clarifying my doubt.
Thank you. So we will go to the next.
One more question is there, then you can proceed.
Sure, please.
This question is from one of our faculty, Mr. Deepak Sadangi. Can any hydrophilic drug will be delivered to lymphatic system in oral dose? If then, which drug delivery system we can suggest?
Okay, so lymphatic is a completely different delivery system. So if you see, I believe that you can refer to many of the journals where it has been very clearly articulated that when somebody is actually targeting to lymphatic drug delivery system, it has to have certain criteria. Okay, the very small molecules also are not suitable for the lymphatic. At the same time, the very high molecular weight are not also suitable. So generally we have seen that if your particle size, like sorry, not molecular weight, I'm just telling the particle size of the product, like if you go to sometimes nano are not very suitable for lymphatic delivery system. Sometimes the micro particles are or the liposomes are very much suitable for the lymphatic drug delivery system. So we have to take care if it is a soluble, water soluble drug or it is not a water soluble drug. That doesn't matter. What it has to be done is that actually as such the drug cannot be targeted towards lymphatic drug delivery system. It has to be developed as a liposomes or microparticles with certain surface modification. Then only we can target towards a microparticle. So I will request Dr. Deepak or Dr. Dilip. I just forgot.
Deepak Sadangi.
Yeah, Dr. Deepak to connect me separately. I can definitely help him, like, because we are currently working on the product on a product which is for lymphatic targeting, and I can certainly help him to make more understand like what are those pharmaco or the physicochemical property or physical properties, mainly the physical property of the dosage form or the particulate particles to be more directed or to be more targeted towards the lymphatic drug delivery system. So maybe it is we are deviating from our current purpose. So I can take it offline.
Okay, thank you. Please go ahead with your presentation.
Yeah. So if you see the case study 3, if you see that this case study is on the itraconazole. Itraconazole, you know, that is a novel antifungal agent, and it's an age-old molecule. It's not. It has been used more than 50-60 years, if I remember correctly. And these are azoles, and if you see this, how it works. This is a pictorial depiction of a fungus cell. And as you know that the fungus cell has a life cycle, and the azoles generally act on the 1,4-alpha demethylase, which helps in converting the lanosterol to ergosterol, and that is helping in making the cell membranes. And the azoles, like other azoles, itraconazole also works on this mechanism, and it inhibits this particular demethylase enzyme, and that's how it restrict or it removes the cell membrane, and that's how the cell death happens. So the major concern currently with respect to itraconazole is it's having a very low viability. So if you see the current gold standard of itraconazole is Sporonox. Okay, Sporonox is the only product currently available which is having a viability of 55%. And I can tell you that many products are available in India, and there are papers, the people have actually done comparative biostudy against Sporonox. Many companies failed to prove bioequivalence with respect to Sporonox, as well as forget about 55% of viability. Many of the formulation has shown even less than 10% viability. So what is happening when actually a antifungal agent is given which is having lower viability, and there are a lot of generic multiples are there which are having even low viability, it creates a threat with respect to fungal resistance. Like antibiotic resistance is there nowadays, the major concern is also the antifungal resistance, and which is a very dangerous situation, because unlike antibiotics, not much research is happening with respect to antifungals, and you can see we have a certain number of antifungals, but looking into this antifungal resistance, many organizations have started actually working, giving more focus towards the development of novel antifungals. So as there is a clear unmet need exist in the market, only the Sporonox is able to deliver the product where there is a 55% viability. But a company in Australia that is called Mine Pharma has come up with a formulation which is called Lozenac 50 mg, which they have proven to be equivalent to, or clinically or therapeutically equivalent to, Sporonox 100 mg. Then even the same company, this Lozenac is only available in Australia. When they went to US, they came up with 65 mg into 2, so 130 mg, which is equivalent to 200 mg of Sporonox. So I will give you a little bit more example why they have come up with 50 mg and 130 mg separate formulation. Why not the same formulation on the next subsequent slides. So we'll start with the first basics about how they have actually done this formulation. So if you see, this is the BCS class 2, itraconazole is a BCS class 2 molecule. That's why it is a very sparingly soluble drug, and that's the major reason that it has a very low viability. It is only soluble in acidic condition, and actually the solubility decreases once the pH increases. And in contrary, the absorption happens in the lower intestine. So the solubility and the absorption is not very much kind of like sync in this kind of thing. So if you see, what they have done is actually the Sporonox is a pellet-based formulation where what they have done is that they have dissolved the drug, and using HPMC as a polymer, they have coated onto the spheres, and it's a pellet-based formulation. And that's how they have actually come up with the formulation which is Sporonox, which is the current gold standard. Okay, so then what Mine Pharma has done, once again. So what Mine Pharma has done, they have come up with a novel technology, and they have kind of like come up with a spray drying as a technology, and that's how they have come up with a product called Lozenac, where they have used a novel polymer, which is HPMC phthalate, which is an enteric polymer, and it is having a pH dependent solubility. So as you know that HPMC is not a pH dependent solubility. It is pH independent, but that's how what happens, the dissolution of drug from Sporonux happens as per the solubility of itraconazole. I told you, hope you remember that itraconazole is a poorly soluble drug, but it has a high solubility in the lower pH at 0.1 SCL, or at stomach pH actually it has a high solubility, whereas when it goes to the intestine, the solubility decreases, and the viability is higher in the like lower part of the intestine. But when Mine Pharma has come up with a formulation which is not made with HPMC, however it has made with the HPMC phthalate, which is a pH dependent polymer. So how, because it's an enteric polymer, the polymer doesn't dissolve at lower pH or stomach. That's how the release of the the release of the drug from the formulation has been restricted at stomach. But when it goes to the intestine, the release is increased, and that's how, as I told, that itraconazole absorbs very well from the intestine. The absorption or the viability has increased. So they have told us that they told that it is a SUBA technology. SUBA is super bioavailability technology. So I will show you some data where you can see that what they have come is that it is a Losinac 50 mg versus Sporonux 100 mg. So you can see this is the Sporonux, which is currently the gold standard. It was a gold standard, but when Losinac has come up with a formulation which is a solid dispersion technology, even you can say that Sporonux was also a solid dispersion technology, but it was moreover, actually, it was a use of hydrophilic polymer and pH independent polymer. But when the Mine Pharma has come, it is a solid dispersion technology where they have used drug is to polymer as 1:1.5, and you can see this picture is a the SEM of only drug. But when it is actually formulated using HPMC phthalate, you can see that the product, the morphology of the product has completely changed, and that's how even we have seen in the XRD that it has been completely converted into amorphous. So it is the drug versus the solid dispersion particles. You can see there is a surface modification, and that's how the it has actually converted into solid dispersions. And what they did is actually, when they actually launched this product in Australia, they have done a comparative biostudy against Sporonux. So 50 mg of Losinac, they have done a comparative biostudy against Sporonux. So you can see when they have actually compared the ratio for all the parameters, so AUC and Cmax, they got about 80%. 79 to 80, 78 to 80%. So if you see, this is not a classical bioequivalence. Like you can tell that if it is not 80 to 125, how the product got actually approved in this country. So what they argued is that for a although the classical bioequivalence is not met, but AUC and AUC versus MIC ratio is more important in comparison to the AUC or Cmax values as such. So what they have told that although the classical bioequivalence is not made, but the desired AUC versus MIC value has been achieved, and it is achieved much more better or much more compliant than the Sporonux, and that's how they could convince the agency to give the approval for 50 mg, and they have got a approval exactly the same indication to Sporonux, which is the Losinac 50 mg can be easily replaceable with Sporonux. So you can see that there are two things happen. One is that there is a reduction in the dose. So 50 mg it is reduced to 50 mg. Second thing is that there is a kind of like increase in the viability, so that the fungal resistance has been reduced with a lower dose, because the and the local side effects, because instead of giving 50 mg, you are now giving instead of giving 100 mg, you are giving 50 mg. So local gastric side effects has been reduced to very lower concentration. So similarly, when they took the product to US, and FDA told that, okay, it's fine that you are showing 50 mg is equivalent to 100 mg with respect to AUC by MIC ratio, but what we feel is that it has to be bioequivalent. Otherwise, we will not be able to give you a level which is exactly similar to Sporanox. So that's how they come up with the formulation. Okay, we are not exactly similar to 100 mg of Sporanox when we are actually comparing against 50 mg. But in this case, what they did, actually they come up with a 60 mg capsule, and where 2 into 60 mg, so 2 capsule of 60 mg, that is 65 mg, which is 130 mg. They have done a study against Sporanox 200 mg, because in most of the fungal infection cases, Sporanox is given twice. Like two capsules it is recommended, so 100 mg into 2. So, and they did a multi-dose study where after 14 days they see the CSS, the steady state concentration, and you can see that the CMX steady state, C trough, and the AUC, you can see the ratio as well as AUC are very much well within the requirement of the bioequivalence. So 80 to 125, it is very well, and that's how they come up with a formulation, two formulation, one for Australia, other one for US. But both the cases, their aim was to reduce the dose, and that's how the patient compliance is improved. The local side effects can be has been reduced. The drug resistance or the antifungal resistance has been reduced. So the next case study is on the levodopa delivery. So you know that levodopa is a potent drug which is generally used for Parkinson disease. Like the Parkinson disease actually is a disease which is regulated through the dopamine concentration in the brain. So if the patient has a fluctuation with respect to dopamine, then the Parkinson disease comes. So we in this case study, I would like to show you that how carbidopa, levodopa, and levodopa carbidopa always is given together, because otherwise you will not get the adequate levels of levodopa in the brain, because carbidopa is kind of a suicidal drug. It gets like it takes, or it does suicide to actually make the levodopa available at the site of action. And I'm going to actually cover like how the immediate release to control release, then the extended release, and the enteral suspension, how this formulation, lot of formulation intervention has happened to take care of different disease conditions. So before we go to the dosing delivery, I would like to just kind of like focus on the disease progression. So if you see what happens to the Parkinson disease, this graph actually depicted that. So if you see, so there are nomenclatures that you can see that it is a normal, Parkinsonism, and dyskinesia. So you can see that generally for 0 to 4 years of the disease period, so it is an early phase, or it is a good year. So if you can see, it is a complete, this is the complete area or the complete regimen we get to kind of like deliver our drug. So in this phase, your IR will easily help. Then what happens, 4 to 7 years of time, it is a kind of like a sort of wearing off a sort of period, where you see you need a very narrow therapeutic window. If you are delivering at this point, then your dosing doesn't work. So that's how you need kind of like a very narrow dosing recommendation. Then what happens when it is 7 to 10 years of Parkinsonism, it is a wearing off with dyskinesia. Like you will dyskinesia as the word says, that involuntarily movement, or you will not be able to actually, the patient will not be able to actually move. It's kind of like your kind of like the movement gets restricted. You are not actually, you are just kind of like a paralyzed. And predictable fluctuation with peak dose dyskinesia. See, if your dose, you are not able to give it within this very short range, you will get dyskinesia. And see, this is the phase where it will get dyskinesia, and above that you will get Parkinsonism. So sorry, this is the phase where you will get Parkinsonism, and when it goes above, it will give dyskinesia. Similarly, when the disease progresses greater than 10 years, then the window goes much narrow. And if you are not able to deliver within this range, then the chances of not treating the disease, which is Parkinsonism, or getting dyskinesia is more higher, and it has a very therapeutic narrow window. So you can see this. So what is the challenges in levodopa delivery? Like, it is more soluble in acidic environment, so you don't get actually more kind of like preference with respect to solubility throughout the GI tract. So it is only soluble when the pH is lower. It is unstable at basic media. Then when it goes from stomach, then it gets more unstable. Narrow absorption window, like it is having a only upper GI tract, it has gets absorbed. So it is like kind of like immediately after the stomach, in the small intestine, it gets absorbed. And it has a unpredictable absorption, because you know that the patient to patient gastric emptying time and gastric emptying is very different. That's why you get a uneven absorption and unpredictable absorption. When the disease progresses and you have a unpredictable absorption, you will certainly get into one of the zone where either the drug is inactive or giving a dyskinesia. Then as the fluctuating levodopa in the plasma level, it impacts the on-off effect. So on-off effect is nothing but whether it is working or not working. So as the drug fluctuates more, you will get that where sometimes it is working, sometimes it is not working. And as the disease progresses, it is the narrow therapeutic window. So you have to be very careful with respect to delivering the drug in a very controlled zone and a controlled delivery system. And there is an inter-subject variability. The drug has a highest inter-subject variability. And so initially, the Cinemet is the first drug, first levodopa carbidopa formulation, which is a IR formulation, which is given. And this only works in the early phase, because it has a demerit of fluctuating plasma level, narrow therapeutic window, and oscillating clinical response. Like, where sometimes it works, sometimes it doesn't work, because you see how the IR formulation works in this case. And then Cinemet came up with a CR formulation, the control list formulation. So when it comes with a control list formulation, because of the narrow therapeutic narrow absorption window, most of the times it is not effective. Because what happens, it is actually once it goes out of the absorption window, there is no further absorption happens, and that's why the patient feels kind of like already the withdrawal symptoms, and having no drug, they will be always actually going back to the disease. And it has a delayed response, because it's a such kind of like a controlled release formulation. That's why the onset of action is also very slow. Then what are the intervention, the formulation intervention people have come up with? I just wanted to give you two examples. One is Rytary. Rytary has come up with a formulation. This is the Impacts. The company name is Impacts in US. They came up with a very novel dosage form, which is a kind of like a ER capsule. So what they have done is actually it's a capsule formulation, and this capsule formulation has IR formulation, IR portion, which is taking care of the onset of action. It has also ER pellets, which is having a very narrow absorption window and having a very stable plasma concentration. And it has SR sustained release tartaric acid pellets. So as the ER pellets releases the drug, at the same time the tartaric acid is also released from this SR pellets, which is actually helping the acidic microenvironment for the product, for the drug. And that's how, as I told in the last slide, you see it has a more solubility in the acidic environment and unstable in the basic media. So this tartaric acid pellet is helping the environment to be acidic. That's how it is helping in absorption as well as making the product stable. And in this way you can see how the product is helping like against the IR formulation. If you see that ER versus IR, it is helping in less chances of failure, less risk of dose jumping, because it's a pellet formulation. Pellet formulation, there is no possibility that all the pellets gets failed at a time. If there is a dose jumping also, there could be a possibility that 1 or 2 pellets get crushed or get ruptured. That's why if it gets dumped, you will not get, the patient will not be impacted highly. And you will get, it helps lower intersubject variability, and it is very reproducible. And you see it's a kind of like a pharmacokinetic data they have given. And you see this ER formulation, the Aicte formulation, is how narrowly it is able to deliver. See, if you see the formulation are able to give 1.7 plus or minus 4 levels of formulation fluctuation. And you, at this point of time, like if you're giving here, the IR is giving 3 plus or minus 1.1. So to give a precise dose delivery system, Aicte is actually doing a very right job. Then when it goes to the last stage of kind of like the Parkinsonism, there you need a very narrow window, and that's how then the formulation has to be kind of like amalgamated with a technology. And this is a kind of like formulation DOPA, where the suspension, the carbidopa levodopa suspension is given through a kind of like directly to the intestine through a bypass. See, you can see this patient has been actually inserted with this device, where the pipe has gone from the stomach, and it has, the end is actually exactly at the lower intestine and the upper intestine. The upper intestine, you can see this this place is the most kind of like suitable position or suitable area where the absorption happens very consistently. And when you need this kind of formulation, like in the very late stage of the disease, you need a formulation which needs to be kind of like very stable and very precise drug delivery system. See, this is the tablet, which is a like oral IR tablet, how zigzag fashion it gives the formulation, whereas this suspension, the enteral suspension, how it is giving a very consistent. I know it is not a very user-friendly solution, but the disease needs that, and there is no other way. So I'm just trying to show you that how the formulation intervention is done to treat the unmet need of the patient. And that's how you can see that in the last stage of kind of like the Parkinsonism, most of the patients undergoes this treatment. Before I go to the next example, which is my last example, if any question, I'll be happy to answer.
Okay, I have a question.
Yeah.
Like in the initial stages of Parkinsonism, they have used this capsule to maintain the pH.
You have to give the tablet. The initial stage of the
next slide.
Okay.
In the case of Aicte company, they have used these capsules. So I just have a suggestion. Can we go for a floating drug delivery system?
So you
where,
can
with one IR portion.
So only thing is that the floating drug delivery systems are till date, actually, if you see, not many commercial products has come, because what happened, actually, it is very difficult to prove the floating of the drug for a longer period of time. See what happens, like although the floating means like you have to target to stomach only, okay, and as you know that there is a housekeeper wave comes for every product, in on everybody, and the stomach contents are completely washed away, and there is always a possibility that your floating drug delivery system may not work until unless I agree there is a company which is working on kind of like foldable floating drug delivery system, where the kind of like the extended release films are made, and they have been folded, or they have been actually kind of like folded into small cylinders and kept it into a capsule, and once it is administered, the capsule actually disrupts, and the sustained release films actually unfolds. When it unfolds, it becomes more than the gastric opening, and because it's a kind of like a floating system, and it's a kind of like a non-erodible kind of system for a longer period of time, there is a possibility to consistently release from that. So I agree, there is a possibility to come up with that kind of formulation. But if you are targeting a floating tablet, may not be. But if it is a floating film, yes, it can help.
Thank you. So you can proceed with your presentation.
Yeah, sure. So my last case study is actually on the reduction of this adverse effect with respect to topical clobetasol propionate cream. So as you are aware that for this clobetasol propionate is a steroid, corticosteroid, and generally the steroids, many steroids are actually kind of like prescribed in many dermatological conditions like psoriasis and atopic dermatitis, and many other skin conditions. And although nobody wants to take steroids, but most of the time actually the severity of the conditions are such high that most of the time actually kind of like the patients are end up with taking steroids. And generally, if you see how the product has come, this Temovate ointment is the innovator for clobetasol ointment or cream. So they have creams and they have ointment as well. So if you see, this is a formulation which is having kind of like 0.05% is the strength. And what this company, which is currently Encore, it is actually developed by Dr. Reddy's. This Impaired is developed by Dr. Reddy's and actually out-licensed to Encore. So currently the company name is Encore, and I was fortunate to actually work on this product, which is kind of like 505(b)(2) application to US, and we have got the fast cycle approval, where we have come up with a formulation which is half the dose, which is 0.025 in comparison to 0.5%, and at the same time we are not compromising with respect to the efficacy. At the same time, we are reducing the systemic exposure of the product, and that's how we are able to reduce the side effects, systemic side effects. So you can see the major ingredient, active ingredient in this formulation, which is here, if you can see, only the propylene glycol can be considered as a kind of like a major penetration enhancer. But in contrary, in Impaired, we have Trascutal and IPM. Both are actually are the penetration enhancer. So the formulation is made in that way that although this Temovate has 0.05% strength, with lower strength, we are able to kind of like release or make available equal quantity of a drug, so the formulation is more permeable. That's why it is going almost equivalent to the 0.05%. So if you see, we have done a clinical study where we have done both safety assessment as well as efficacy assessment. You can see this is a comparative systemic exposure. So it is a 0.025%, where the systemic exposure is only 56.3 picogram per ml in comparison to clobetasol propionate cream, which is the Temovate cream, which is 152.5. So you can see that almost 1/3 reduction in the systemic concentration. So our formulation is able to penetrate within the layer of the skin. It is not going to the systemic circulation, where this product is required for local use. It is not required to be absorbed into the systemic circulation because it is not a systemic drug. Okay, so that's how we are able to restrict our formulation into the skin concentration, and we are releasing the drug into the systemic circulation almost 1/3 of the conventional clobetasol propionate cream. So if you see, what we have seen, that generally the safety of a corticosteroid is kind of like evaluated, because all the corticosteroids are responsible for suppression of HPA axis. HPA axis is the hypothalamus pituitary adrenal axis, which has a lot of functions in our body, and if it gets suppressed, like a lot of dysfunctionality will happen in the body, and sometimes the person may get anaphylactic shock also. So if you see the incidence of the HPA axis suppression, you can see that it is significantly reduced, 36% compared to 11% only. So we have done a study, HPA axis suppression study, and we could see that almost 1/3 or more than 1/3 reduction in the HPA axis, which depicts that our formulation 0.025 formulation is much more safer compared to the 0.05% formulation. And you can see that the assessment of efficacy, we have done the efficacy with respect to the reduction in the psoriatic patches, and there is a investigator global assessment score is there. So you can see in these pictures, all the patients, like this patient has been started with the IGA score of 3. So the higher the score, higher the disease condition. And after 3 days, then followed by 10 days and followed by 15 days, you can see the reduction in the disease condition. And it started with IGA score 3, and it has at 15 days it has reduced to IGA score 2. And eventually the people has been seen, like even another example, you can see this is also started with IGA score 3, and after 13 days it has reduced to IGA score 1. So it shows that it has a equal efficacy as well. And again, some example, like if you can see, this is how a person hand was looking like with plaque psoriasis, and after 15 days of use, you can see that almost there is no visible sign and symptom of psoriasis in there. Similarly, another example where a patient is having a psoriatic condition over here, and you can see that after 2 weeks it has completely almost vanished. So this is how we are able to actually come up with a formulation which is having less side effect with uncompromised efficacy. So these are some examples I wanted to show you, which I believe you are able to understand, like how the differentiated formulation is being developed, and how we are able to understand what are the unmet needs, and how we are actually working around the formulation to come up with a formulation which is having equal efficacy with uncompromised efficacy, and much reduced side effects, much more patient compliance, and we are trying to kind of like reinvent the old molecules and coming up with a new kind of like new formulation intervention. With the new formulation intervention, we are able to actually come up with a very superior dosage form. That's all from me today. I'm open for any questions or queries. Dr. Niranjan, I think you are on mute.
Thank you, Mr. Arnab Abhijit Chaudhary, for your elaborative presentation on differentiated formulations. Really, it was one of the most practical presentations where you have explained the concept of differentiated formulation with case studies, and we got an idea how the industry professionals approach in developing a formulations. So thank you very much. Now the session is open for question answer. So any question is there from the participants, either you raise your hand or you can type your question in the chat box.
So one thing, by the time like people are actually getting ready for the questions and they're typing questions or they are ready to ask questions, I just want to also invite all of you to kind of like come up with this kind of novel concept, and we will be very much open to collaborate with you to take this product or the concept forward, and kind of like doing collaborative research. So we are always in search of organizations, institutions who are actually working on differentiated formulation, and we are very much open to kind of like collaborate and kind of like take the formulation, and we can actually work on kind of like industry institution relationship model.
Thank you very much for this offer. Definitely, we would like to have some collaboration with you, and I request all our faculty members and participants to come out with innovative ideas so that industry will accept our proposal and we can do something for the betterment of society.
Yeah.
So any question from the participants? Okay. So I think during the session only many questions were discussed. So thank you, Mr. Anup Abhijit Chaudhary, for accepting our invitation, and please accept a e-certificate on behalf of Roland Institute of Pharmaceutical Sciences as a token of appreciation.
Thank you. Thanks a lot.
And
thanks a lot for
forward.
Yeah, please go ahead. Please go ahead.
We are looking forward for your cooperation in future also.
Sure. I'll be always there. So it is my responsibility as an alumni to give back to my organization, institution, and definitely I'll be always there to collaborate and whatever best I can do, I'm always ready to do that.
Thank you very much.
Thank you for your invitation. Thanks the organizers and the sponsors for the valuable interaction time, and thanks all the participants for your patient listening. Hope I could actually kind of like contributed to your learning, and I have used the time judiciously. Thank you.
So there is a last question. Just let us complete with that question.
Sure.
One of the question is what are the limitations of this differentiated formulation?
So sometimes limitations, if you see, if you are, it all depends on the kind of like the depth of the innovation. And in most of the cases, we are able to convince the regulators with minimal studies, so that we are able to reduce, we are able to keep a control over the cost of the development. But in some cases, like where we are going very deep into the research, and the agencies, most time the regulatory agencies comes back to us heavily, and they tell that you have to do a lot of battery of studies. There is a limitation, because almost it goes to kind of like the cost of the research as well as the timelines of the research goes very near to the NCs. So there is something which we need to be keeping a fine balance, so that we are not going beyond a certain year of studies or certain number of studies. Otherwise, the commercial viability of the product doesn't become very viable.
Thank you very much. So this AICT sponsored short-term training program is coming to an end. Thank you all the participants for your support and cooperation.
Thank you.